Hey friends, in this video we'll discuss about the absorption of the drug. So first we must know that what does it mean? Absorption is a transfer of the drug from the site of administration to the bloodstream. There are many factors that may influence the absorption of the drug, but the major one are the route of administration. So the rate and efficiency can be evaluated from the route of administration. For example, the IV delivery, the drug reaches to the bloodstream with its maximum concentration, while through the oral route the drug may face the harsh environment of the GIT. It may faces the different pH levels and the different enzymes, so it may get metabolized, and in this way the bioavailability also decreases. So in this we will discuss the transport of the drug from the GIT and the effect of the pH on the drug absorption. Practically, the drug should be able to reach its intended site of action. In many cases, the active ingredients are administered as such because they are readily lipid soluble, and thus they produce their effect so easily. But sometimes the drugs are given in some chemical precursor form where the active ingredient is insoluble in lipid membranes, and in this way the chemical precursor are converted into the active form by biological process. So this precursor is known as prodrug. Sometimes the drug are applied directly to the target tissue, for example in case of topical applications, but sometimes we need to administer in one compartment of the body and then it reaches to the target site after passing through different barriers. For example, if the drug is intended to produce its effect in CNS and we give it orally, then what kind of the barriers we expect to come in the passage of the drug will be the membranes of the, first of all, the gut wall, the membrane of the capillaries that perfuse the gut, then the blood-brain barriers, then the membranes of the capillaries of the brain, and then finally, after bringing about its effect, the drug should be eliminated. So while discussing the transport of the drug, the first mechanism is permeation, and drug permeation has different mechanisms. Passive diffusion, that mostly occur in aqueous or lipid medium, but the active process that play a very important role in the movement of those drugs having too large to diffuse readily. Drug vehicles also play a very important role in their permeation. For example, if we formulate the active ingredient with liposomes, then this would also facilitate its transport. The second one is a fixed law of diffusion, and the third one is the Henderson-Hasselbalch equation. So first we will discuss in detail the permeation. Among permeation, the first one is the aqueous diffusion. So let's discuss that where this type of diffusion occurs. It's found to be present in a larger aqueous compartment of the body, for example interstitial spaces and cytosol, across the epithelial membrane tight junctions, and in the endothelial lining of the blood vessels. So there are also some other tissues in our body, for example the capillaries of the brain and testes, that lack the pores, and this also permit the aqueous diffusion. This kind of diffusion most importantly allow the drug to move from higher concentration to the lower concentration, which means downhill movement, and this would be better explained by the fixed law of diffusion. Drug molecules, when moved from higher concentration to the lower concentration, which means that this kind of diffusion is not saturable and they do not involve any kind of a carrier, they have a low structure specificity. What are the limitations of this aqueous diffusion? So let's see. Those drug molecules which are bound to large plasma proteins, for example albumin, do not use this kind of diffusion, and those drug molecules which are charged, their diffusion through this passage can also be influenced by the electrical fields. the second one is the lipid diffusion, and it is the most important limiting factor for the drug permeation because large number of lipid bilayers separate the different compartments of the body. As we know that the lipid barriers separate the aqueous compartments, so the lipid aqueous partition coefficient of the drug will determine that how readily the molecules move between the aqueous and the lipid media, especially in the case of weak acids and weak bases, which carries the charge. The ability of the drug molecules to move from lipid media to aqueous media or from aqueous media to the lipid media, it depends on the pH of the medium because charged molecules always attract the water. So this would be better explained by Henderson-Hasselbalch equation, which we will discuss later. The third one is the special carriers type of permeation. As the name indicates, this type of permeation includes special carriers that are present for those substances that play a very important role in the cell function and for those substances that are quite large in structure and are unable to absorb from the lipid through passive diffusion. For example, different amino acids, peptides, etc. So these carrier molecules bring the movement of the particles by active transport or either by facilitated diffusion. So this kind of diffusion or this kind of movement is saturable and it is energy dependent because in this type of diffusion the molecules move against the concentration gradient from their lower concentration to the higher concentration. In most of the cases the energy is driven by the hydrolysis of the ATP. So different cells contain less selective membrane carriers and they are specialized for expelling the foreign molecules, and one large family is of ATP is known as the ABC family that binds to the ATP, and this family includes the P-glycoprotein and multi-drug resistant type 1 transporter that are found in brain and testes and other tissues and in some drug resistant neoplastic cells. So they play a very important role in expelling the foreign particles from the respective tissues. The last one is endocytosis and exocytosis, and this method is reserved only for those large molecules that can enter the cell only by endocytosis, and this process involves the attachment of that large molecule with some cell surface receptors, and after attachment these large molecules are engulfed and then carried into the cell by pinching off of the newly formed vesicle inside the membrane. The substance can then be released into the cytosol by breakdown of the vesicle membrane. The process is basically responsible for the transport of the vitamin B12 that is complexed with some intrinsic factor across the wall of the gut into the bloodstream, and similarly the iron is transported into the hemoglobin synthesizing red blood cell precursors in association with the protein transferrin. Specific receptors therefore should be present on the cell surface so that the large molecules can interact with those receptors. The exocytosis is simply reverse of the endocytosis, but this is responsible for the secretion of many substances from the cell. For example, many neurotransmitter substances are stored in the membrane-bounded vesicle in the nerve endings in order to protect them from metabolic destruction in the cytoplasm. So, when there is an appropriate stimuli, the membrane-bounded vesicle attach themselves to the inner membrane of the nerve ending, and this causes the expulsion of the content into the extracellular space. So this was about the exocytosis. Fick's law basically tells us about the passive movement of the molecules down a concentration gradient. So in this equation, C1 is the highest concentration, C2 is the lowest concentration, area is basically the cross-sectional area of the diffusion path, Permeability coefficient is a measure of the mobility of the drug molecules in the medium of the diffusion path, and thickness is the length of the diffusion path. So in the lipid diffusion, the lipid aqueous partition coefficient is a major determinant of the mobility of the drug because it tells that how readily the drug will move from lipid to aqueous medium or from aqueous to lipid medium. The Henderson-Hasselbalch equation tells us about the absorption of the ionized form of weak acidic and weak basic drugs. Lipid diffusion depends on relatively high lipid solubility and ionization of the drug that reduces their ability to permeate the membrane. Keep in mind that always the charged particle will not be able to cross the lipid membrane, while the neutral particle will easily be able to cross the lipid membranes as it is uncharged, free of charge. So what are the weak acidic and weak basic drugs? Weak acidic drugs can be best explained as a neutral molecule that can reversibly dissociate into an anion and a proton, while the weak basic drugs can be defined as a neutral molecule that can form a cation by combining with a proton. So keep in mind that the protonated form of the weak acid is a neutral form, and that's why it is more lipid soluble, whereas the unprotonated form of a weak base is neutral form. According to the law of mass action, these reactions move to the left in an acidic environment while moves to the right in an alkaline environment. The first equation is of the Henderson-Hasselbalch equation, and this applies for both acidic and basic drugs. Let us take an example of weak acidic drugs. For the weak acidic drugs, we have shown another image of neutral aspirin that reversibly converted into aspirin A9 plus proton. So according to the above equation, the lesser the pH, which means that more acidic environment in the stomach, will causes the equation to moves towards the left, and in this way more neutral form or more protonated form of the drug will be available in the stomach, and in this way more drug will be absorbed from the stomach, and this is for the weak acidic drug in stomach. But when we discuss the weak acidic drugs in intestine, where the pH is slightly basic, the equation will move towards the right, which means more anionic form of the drug will be available, and in this way lesser will be the drug absorbed from that media. So for our convenience, we can say that this follows the like dissolve like phenomena, which means that acidic drug will be absorbed from the acidic media and the basic drug will be absorbed from the basic media. Let us take another example of weak basic drug. So in case of weak basic drugs, when these drugs are present in stomach, where the pH is more acidic, the equation will move towards the left and more ionized drug will be present. In this way, lesser the absorption of the weak basic drugs in stomach take place. While in the same drug enters the intestine where the pH is basic, the equation will move towards the right and more neutral form of the weak basic drug will be available for the absorption, and more absorption will be take place. The Henderson-Hasselbalch equation, we can find out that the way how to accelerate the excretion of the drugs. As we all know that almost all drugs are filtered in the glomerulus, and if the drug is in a lipid soluble form, during its passage down the renal tubule, significant fraction of the drug will be reabsorbed by the simple passive diffusion. So if the goal is to accelerate the excretion of the drug, then we need to change the pH of the urine. For example, the weak acidic drug will be excreted more faster in alkaline medium, because in that case, in that medium, the drug will be in ionized form, and in this way the ionized drug will not be able to reabsorb or cross the lipid membrane of the glomerulus. So same in the case of weak basic drugs, which will be excreted faster in acidic media. So most of the weak basic drugs are amine containing molecules, and the nitrogen of the neutral amine has basically 3 atoms associated with it plus a pair of unshared electrons with which it can bind to different molecules and forming the different structures. So the 3 atoms associated with the nitrogen may contain 1 carbon or a carbon chain and 2 hydrogen forming a primary amine, or it may contain 2 carbon chains with 1 hydrogen attached to the nitrogen forming secondary amine, or the nitrogen may contain 3 carbon chains forming the tertiary amine. While there is another type of the amine molecules, which is the quaternary amine, in which the nitrogen has no lone pair of electrons and all of its electrons are bound to carbon chains, and this form of the amine remain in charged form, while the primary, secondary and tertiary can bind to the proton and form the protonated form of the drug, and in this way their lipid solubility varies with the pH value, but the quaternary amine that always remain in poorly lipid soluble charged form. So this was all about the absorption, permeation, or we can say that the transport of the drug that I explained in this video.